The basic unit of the nervous system is the neuron. The majority of the neuron's organelles are located in the cell body. The highly branched dendrites on the neuron receive signals from other neurons. The electric signals are integrated into the base of the axon before an action potential is generated. The neuron has a single axon through which the signal or action potential travels. A lipid myelin sheath insulates the axon and keeps the signal moving down in one direction. Between each unit of myelin sheath, the ion-gated channels at each node conduct the signal. Each unit of myelin sheath is called a Schwann cell. The axon's other end branches off into the axon terminal. The terminal sends information to other cells at a junction called the synapse. The part of the branch that makes up this specialized junction is called the synaptic terminal. Here, chemical messengers called neurotransmitters pass information from the transmitting presynaptic cell to the receiving postsynaptic cell. Action potential depends on hyperpolarization or depolarization of the neuron to reach its threshold. Since the opposing charges act as a source of potential energy, the charge difference on either side of the neuron cell membrane is called the membrane potential. The resting potential of the neurons not transmitting signals is typically between -80 and -60 millivolts. Potassium and sodium ions play an important part in creating the resting potential. These ions both have an individual concentration gradient across the plasma membrane. In most neurons, the potassium is higher on the inside and the sodium is higher on the outside of the cell, and their concentration gradients are maintained by the sodium-potassium pump. This protein pump hydrolyzes ATP to gain energy and actively transports sodium into the cell and potassium out of the cell. The pump transports 3 sodium per 2 potassium ions, resulting in net differences. Ions that diffuse through the channels carry units of electrical charge, a net movement of which would generate a membrane potential. At rest, the neuron has a membrane potential of -70 millivolts. When a stimulus is applied, the neuron increases in potential, crosses the threshold, and rises in potential about 30 millivolts. After reaching the maximum potential, the voltage falls below its resting potential at first and then rises slightly to the rest position. Botulinum toxin, or BTX, is a toxin produced by the bacteria Clostridium botulinum. BTX is a type of synaptic vesicle release inhibitor and prevents acetylcholine release at the neuromuscular junction. BTX is responsible for botulism, a rare but lethal form of food poisoning, but in controlled doses, the toxin can relax the muscles, get rid of wrinkles, and prevent spastic muscle activity. When BTX is introduced into the bloodstream or muscle, the toxin binds with receptors on the neuron membrane. Its light chain is then split from its heavy chain and taken into the neuron via endocytosis. The light chain of BTX then cleaves the snare proteins that help synaptic vesicles fuse with the neuron membrane in order to release its neurotransmitters. In this case, BTX affects the release of acetylcholine, the neurotransmitter responsible for muscle contraction. Without the ability for the vesicles to fuse and release acetylcholine, the affected muscle cannot contract. Effects of BTX include loss of facial expression, nausea, eyelid laxity, ptosis, and death.